the like shoot, I forgot what it was, but yeah.
Yeah, it's a general principle that the simplest explanation is more probable. You know, because you can explain a lot of things by making a very elaborate model as to how it occurs. But if there's another model that can explain the same information in a simpler fashion, then it's more likely that the simpler fashion is well, to be preferred. Okay.
And does simpler mean like less
complicated? Less complicated, yes.
So like that would be like less levels going.
Yeah, and so in this case, when I'm talking about phylogeny, parsimony and phylogeny, we're looking for the arrangement of the species that explains our data, which in this case is our DNA data, with the fewest number of steps. Where I have here one step, whereas over here it requires, this particular topology requires 2 steps. This is simpler. And likewise, the complete tree is down at the bottom. The one with 6 steps is more parsimonious than the one with 7 steps. I see you thinking.
Yeah, how is the one on the right 7 steps and the one on the left 6 steps?
Okay, if you look up here, this is showing all the 3 characters, or let me see here, we have 4 characters, 4 different characters and where they had to occur. Now here, T, or number 2, had arisen just once, okay? But 4 has to rise twice independently. 3 had to rise twice independently. And so if you count, this here has 1, 2, 3, 4, 5, it should be 6. It must be leaving off. Let me see, this has 3, 2, it doesn't have 1 on there. That's what's missing. Okay, so this tree down here is superimposing this tree and this tree on the same branching pattern. And so here, this is 1 and 2, right there occurs just once, whereas 3 had to arrive here and there twice, 4 had to come up twice. And so this is 1, 2, 3, 4, 5, 6. Whereas over here, to explain it, it's a shame they didn't put the numbers on here. 1, 2, 3, 4, 5, 6, 7.
So each tick is like another step.
Yeah, yeah. That's what we're talking about, parsimonious. The one that requires the fewest number of events to explain the information, that's more parsimonious.
Okay.
Okay. All right. So what does, once again, phylogeny have to do with classification? Linnaeus provided this particular hierarchy, okay? Species, these are populations, interbreeding populations that are gathered into genera, several genera, as families, orders, classes, phylum, kingdom. I still want you to know these basic groups, although, because we will refer to them once in a while. Mostly it's the order, family, genus, and species that really is more useful. When it comes to deeper phylogenies, a lot of times I don't care too much about the kingdom and phylum and class type of deal. But nonetheless, the relationship between phylogeny and classification. Let's say that this is a phylogeny, and let's say that all these taxa A through L happen to be genera, okay? And this is, we want to put together monophyletic groups, and so we can group A and B. And of course, the definition of a monophyletic group is that all the taxa that are descended from, they share a recent common ancestor. And so here at the node, get my pen so I can draw. That's not it. Where is it? It's right here. That's the node. And that's the most recent common ancestor of A and B. And therefore, this is a monophyletic group because it includes A and B and all of the descendants of that most recent common ancestor. We can also do that with the next clade, C through E, and put that together. And F and G and their most recent common ancestor, H and I, J is by itself, and K and L. Okay, and let's say, since these are genera, then that grouping is a family. And so A plus B is a family. C plus D plus E is a family. F and G is a family. And then we can put families together in their most recent common ancestor. And so this whole clade that includes A through G, the most recent common ancestor for it is, of course, further back in time. And likewise, the other clade with its most recent common ancestor right here, H through L, then we have orders. Now we have two clades here, and we can, went the wrong direction, the red line, put them together as a class. Okay, now, so this is the relationship between the phylogenies and monophyletic groups, how we put them together in a Linnaean classification. Yes, Alquicin.
I just had a question about C, D, and E. So we could put D and E into a monophyletic group, but we can't put C into one by itself because it has to include D and E, right?
That's right. That's right.
Okay,
You could do D and E, but if you left out C, then it becomes a problem.
So wait, I have a question.
And
What was D and
actually could. No, you could. You could put these together as a monophyletic group, but you couldn't include this ancestor in it, that group.
so what would that monophyletic group be? Because it wouldn't be a family, because that would have to have C, right?
Right. You see, that's the problem with the Linnaean classification, is that we have so many branches. What are you going to call it? Family, infrafamily, superfamily, subfamily, and we run out of prefixes. Okay, so this is still a bit useful, but I prefer what I've already introduced in exercise 2 or exercise 3 as a lineage classification. Just forget about the categories. Just put together the order from broadest, most inclusive to the more specific groups, the clades. Just name the order of the clades. Okay, and that's where I'm going. All right. So, but we have something else to discuss here. What do we do about fossils? Okay, here I have species X or genus X. All these A through L, these are living organisms that we have today. All right, but X is extinct. Where do we put it? And what about Y? Y is more closely related to B because it shares a more recent common ancestor between B and Y than what B and A have further back in time. Y can belong to this genus, but it's extinct. And so what do we do with it? How do we talk about it? Well, we need to refer to two different classificatory phylogenetic terms here, referring to the crown group and the stem group. The crown group is based on a clade that includes living organisms. Okay, give an example of this, mammals. Okay, we have mammals, we have some mammals that are extinct too. Okay, but the crown group, we would take the, if we had a phylogeny here, let's say that H happened to be a duck-billed platypus. Okay, these are egg-laying mammals, and we believe them to be, they retain some rather plesiomorphic characters, and therefore we believe that they branched off from the placental mammals and even the marsupials really some time ago. And so the crown group would include the duck-billed platypus and all the rest of the living mammals. Okay, that is the crown group. It's based on the living organisms. Okay, like crown group Aves would include a robin, and let me see, what are the more primitive emu, okay, and their most recent common ancestors. That captures all of the living birds, but Archaeopteryx, a Cretaceous bird, is extinct and lies outside of that group. It doesn't belong to the crown group clade. So X here is not inside of a crown group. So this group here that includes H through L, that is a crown group. And it does mean that there's probably members within the crown group that are extinct. Okay, still belong there. But X lies in what is called the stem group. The stem group is anything that connects the ancestor of a crown group with the ancestor of a broader crown group, okay? So if we go down to this ancestor, well, our crown group includes A through L and all the fossils that belong therein. When we go to this bigger crown group for a bigger clade, then X becomes part of the crown group. But if we're just only considering H through L, that particular clade, then X is a member of the stem group. Okay.
So that just means it's on the stem reaching that group, right?
That's right.
Okay.
That's right. It's an extinct member, and it's it left no living relatives, and it's not within a crown group, but it connects crown groups. Okay, this will make more sense when we get a look at the ancestry of mammalia, mammals. That's on a totally different amniotic branch than what turtles, lizards, dinosaurs, and birds belong to. There was two different branches, but we have all these fossils of mammals, and a lot of times in older textbooks, they're called reptilian, mammal-like reptiles. Okay, but they're not reptiles. We really need to quit saying it like that, but that's how they're called. They have a lot of reptilian-like characters, but we see a gradual movement in their morphology toward mammals, and they really belong on the mammalian branch, but they're not really mammals. They're stem groups, okay? They belong to the stem group Mammalia. But when we're looking at the group Amniota, which would include both the lineages that goes to turtles and other reptiles, including birds, and the one going to mammals, then those stem groups are part of the larger crown group. Yes, Alcacin.
Sorry, one last question. Would Y be a stem group of the genus B?
Yes.
Okay.
Yes. Yeah, it would be. Yeah, so if we have, because here it's implied that there's several species, a lot of species in B, right? Because that's a genus. And so, yes, the most recent common ancestor of B would, and the species of B would be right here. And so under those circumstances, Y would be in the stem group.
But we couldn't say a genus could be a stem group of a species, right? We can't say that.
No.
Okay.
Yeah. Okay. All right. So as I said, lineages. We forget about, and I'm going to be using lineage quite a bit, because as we get into the diversity, we're going to be looking at a lot of clades and looking at the phylogeny. And really what is nice about this lineage concept is it gives, you get the phylogeny just from the way it's classified. And we don't have this problem with trying to think of, is this a family? Is this a order? Is this a phylum? Or whatever. Nonetheless, I will mention that on certain groups, how they're treated in the Linnaean classification. But this is your classification, and it's not even complete, okay? Eukarya, the broadest part, and that's a domain. And within it, you belong to the kingdom Animalia. And it's hard to say what these ranks are in here. You know, in a Linnaean classification, we'd have to say domain Eukarya. And what do we do about amorphia? We'd have to call it, what, a subkingdom, or no, a superkingdom, or a subdomain, or something like that. And that gets to be very, very, very cluttered. Furthermore, a lot of individuals in the past wanted to equate kingdom Animalia and kingdom Fungi, for example, as if they're somewhat equal, but they arose at totally different evolutionary times in the past. And so I like this Linnaean classification, and it gives the phylogeny. So this is, and you might look at this and say, oh God, am I going to have to learn all those words? Most of them you will, yeah. And don't worry, don't fret about it, because I've been through this for 15 years now, or this particular concept. And students do okay. You know, we're going to learn it gradually. So I'm not going to ask you any of these taxon names in the immediate future, but we'll gradually work our way down the tree. But this is your classification. So first of all, this is a simplified phylogeny of Eukarya, okay? And Eukarya or Eukaryota. These are two words, they're synonymous, they mean the same thing. This is the domain of eukaryotes. And within the Eukaryota, there are 5 different groups which sometimes are called supergroups, but we belong to the one called Amorphia, which is this branch over here, right? And I have it written there, Amorphia. Eukarya is down here. All of these are eukaryotes, but only this branch, Amorphians. And within the Amorphia, we belong to Animalia. Now, I'm leaving out some things because there's a special name right here too that we'll be using a little bit later on, but I didn't put it up here in this classification. And so, you know, fungi used to be classified as a plant once a long time ago. But you notice here with our phylogeny, our plants are way over here. And the most recent common ancestor of fungi and land plants is way down here. And so this would include Animalia as well. Emily.
Professor, is Uniconta the same thing as Amorphia?
Say that, yeah. Do you guys have that in your? Oh yeah. This was just changed with the last edition of this textbook. Yeah, so this here, Amorphia, does equal Uniconta. And Uniconta, it's a shame they changed the name. They changed it for a very trivial reason, but I'm going with what the textbook says. And also Wikipedia will point out that this is now an invalid name. It looks like I didn't change it in your notes. Okay, so whenever you come across the Uniconta, just change it to Amorphia. Okay, and I'm going to write it down so I make sure I change it in the notes. Or else I'll do this again next year because I already did it last year. All right, and so if we go in a little bit down within the Animalia, Eumetazoa, that's where we are. Not all animals are eumetazoans. And then within the eumetazoans, the branch Bilateria is what we belong to. And then within the Bilateria, we belong to the branch Deuterostomia, and so forth. And we will walk it. I'm not going to go through all this, but we'll walk through a lot of this in the next several weeks. Okay. All right. So any questions? Any slide that you need to see?
So like if you were to ask us to draw a phylogeny of Animalia, we would draw it just like that, going from Animalia as the bottom and then up.
That's right.
Okay.
Yeah, because there's various ways to draw phylogenetic trees, and a lot of textbooks or papers like that have them is go here. I prefer something like this, but the textbooks a lot of times like to have these types of trees.
And then to that we would.
They're okay, but I just don't like them myself. I'm prejudiced.
And then to that we would add the apomorphies if we were asked for them, right?
That's right.
Okay.
Yeah. So you'd have to put the apomorphies on the branch that they belong to, and so forth. Exactly. Okay, so then let me put that down, and so we've looked here now at the general principles of evolution, from microevolution to, second here, I've got to fix this, macroevolution and speciation, and how that brings about counts for the distribution of life on Earth. So now what I'd like to do is look at just not in great detail, but in enough detail to give you some idea of what we think we know about the fossil history of the Earth, because this has been the consequence of evolutionary change. And so this third topic is looking at the history of life on Earth. So first of all, go through chronologically. So I'm going to go from the oldest to the most recent, and of course we're going to talk about just only the major events. But first of all, the Earth, we believe it's 4.6 billion years old, and that is, but still the oldest rocks that we have on the Earth that has been found are only about 3.9 billion years old. And the reason we think that the Earth is about 4.6 billion years old is because of meteorites. Okay, certain types of meteorites, almost all of them that have fallen to Earth, we believe them to be some of the remnants of the early formation of the solar system, and they are 4.6 billion years old. Now, why can't we find 4.6 billion year old rocks on Earth? It's because the Earth's surface is very dynamic. It has this erosion, mountain building, and tectonics with everything moving around, and a lot of this rock could have been destroyed, or it is buried into a place where we don't have access to it. And so we haven't found those types of rocks. Now, it is believed, and this was not put up by biologists, but by earth scientists, geologists, that they believed that the early Earth's atmosphere was a reducing atmosphere. It did not have oxygen in it, okay? And it consisted of H2S, sulfur, and ammonia, methane, cyanide, among other things. It was pretty much in the atmosphere. Carbon dioxide too, that is carbon dioxide, and carbon monoxide as well, but not a whole lot of oxygen. In fact, most of the oxygen on Earth was, and still is, locked up in minerals, okay, in like silicon and so forth. But today, our atmosphere is about 21% oxygen, and it makes the Earth a very rare planet in our solar system, having so much oxygen gas. So where did this oxygen come from? Well, we'll get to it a little bit later. So with no oxygen in the atmosphere, there was also no ozone layer. Now, ozone layer, it's way up in the stratosphere, and it's O3, okay? And it absorbs UV light. And in fact, the sun puts out quite a bit of UV light. Some of that UV light, ultraviolet radiation, does make it down to the Earth. That's what gives you a sunburn when you're out in the sun. But the ozone layer absorbs a lot of the UV light. The UV light would be much, much stronger. And if without that ozone there, and cancer rates would go up pretty high. Now, what was interesting is two individuals, Stanley Miller and Harold Urey. In 1953, they did a particular type of experiment. They proposed, when given the proposal of what the early Earth's atmosphere happened to be like. And so in their laboratory, this is what they did. Now, this up here says 4.6 billion years ago. But I think the more recent numbers for 4.6 instead of 4.5. They put together ammonia, methane, water, molecular hydrogen, some of the components that was believed to be in the early Earth's atmosphere. And it was also believed that during that time, there was a lot of lightning storms, okay? So they put in an energy source, such as electrodes here and spark, to simulate lightning strikes and even high energy input from UV light. And eventually they would, after a week or so, they took a sample out of the condensate here and wanted to see what was in there. So this was simulating early Earth's atmosphere as far as what they believed. And what they would find is that when that isolate material, they would find some amino acids that had formed spontaneously, and nitrogen bases, simple sugars, and the basic building blocks of life. Now, what's interesting is some of these amino acids some of them are the amino acids that are found in life today in proteins, but there's also a lot of other different amino acids that are not found in proteins. And furthermore, these had both about equal numbers of L enantiomers and R enantiomers, whereas in living organisms, it's only the L enantiomers that are populated in the proteins. Furthermore, they could get these to polymerize if they put them on clay particles and evaporated some of the water. Then some of these amino acids would form short peptides and so forth. Some of the simple sugars would form not full polysaccharides, but something along that line. So what this says is that there must have been a early chemical evolution on life, which we really don't know a whole lot about. And this has been studied now for a good 50 years or more. And cell membranes, those polypeptide or phospholipids, they were also in this mixture. But from the formation of these early chemicals, organic chemicals, to that of a cell is a big question mark. What events happened, we don't know. But we do believe that that life on Earth evolved from, first of all, chemical evolution, and eventually we came to first living cells. We have strong evidence based on the ways in which proteins are made and DNA is stored and used to store information. We have reason to believe that life only occurred once, or if it did evolve more than once, then still the life that we have that succeeded came from one ancestor. But these are very open questions. Yes, Alkasim.
I may be wrong, but is that why life started in like the water, because the atmosphere has no oxygen?
No, the earliest forms of life, in fact, among the bacteria, the groups that are, we believe, are pretty ancient in their biology are anaerobic. Oxygen, atmospheric oxygen, is actually poison to them. And so life, there are living forms that oxygen is poisonous to it. But also having oxygen and being able to split water in photosynthesis was a big boon for multicellular life. And probably multicellular life would not be possible without molecular oxygen to drive higher levels of ATP production. Right now, it's not real certain where life actually started. Right now, there's a hypothesis that it was really in deep thermal vents in the ocean. That's also in a place where there's not, there isn't any oxygen, but there's a lot of sulfur compounds that are coming out of those heat vents that can be used as an energy source instead of water for splitting water to like happens in photosynthesis. That appears to have come later.
So did the life itself contribute to the oxygen in the atmosphere?
Indeed, it did.
Okay.
Yeah, we believe that the oxygen in the atmosphere is due to the evolution of oxygenic photosynthesis.
Okay, thank you.
Yeah, because there's other types of photosynthesis in bacteria that last semester we really didn't go into, but they don't split water to create oxygen as happens in oxygenic photosynthesis in cyanobacteria. We have fossil cyanobacteria a long time ago. Okay. All right, now another hypothesis here too is the strange, the idea that DNA was not the first storage molecule, hereditary molecule, because there's this sort of chicken and egg type of problem here with DNA and protein. Indeed, the DNA encodes the information for protein, but the factories that are used to make that protein and interpret the information from the DNA is protein. So how can you get around all this? And so it's believed that maybe the first forms of life were based on RNA, because we have some remnants of RNA within living organisms that play some very interesting roles, not just only in information processing, but some RNAs are also catalytic. And so there could have been an RNA world first before the real evolution of having proteins take over doing a lot of the hard work, okay? And so this is theoretical. And so there may have been, but however it started, we still have reason to believe that it happened only once. And once we get a living cell, then we can start talking about things a little bit better. All right. And so that's what I'm going to concentrate on. This question of how life really arose is still, nobody knows for sure. That there's just only hints of it, because since they we can see that under a reducing atmosphere, that is an atmosphere free of oxygen, that organic molecules such as amino acids, simple sugars, etc., they can spontaneously form, okay, which are the building blocks of life. Now that wouldn't happen today because oxygen is very, very reactive, and those molecules wouldn't last very long. Okay, but so what evidence do we have for life? from fossils. And so I need to talk a little bit about fossils. What are fossils? The remains of ancient life or its activity. Now, one type of fossil that's very, very common are called imprint fossils, and they show up in sedimentary rock. And so here we have sediment that's being washed into some basin. It could be an ocean basin, it could be a lake, it could be some other low-lying area where water comes in, buries the dead animal or plant or whatever it happens to be, and entombs it. Now, most of dead animals, if you look at what happens out here in your real world, is that they almost never have a chance to be fossilized. Now, there's other living forms, bacteria, other animals that come along, eat it up, tear it up, and destroy it long before it ever gets entombed in sediments. But every now and then that can happen. And so once we get it entombed and it forms into rock, because first of all, it's sediment, and then the clay or sand particles, are then cemented together, for example, with calcium carbonate that happens to be in the water, it forms a cement, and we have sedimentary rock, and as the organism decays, it leaves an imprint. And so this is a Paleozoic imprint of a dragonfly, and it's not a type of dragonfly that's around today. Okay, in fact, some of these dragonflies from the Carboniferous era happen to have wingspans of about a meter. Okay, they're pretty big. And here is another imprint fossil of a fish. And I have other fossils. These fossils come in different forms. We can also have trace fossils. Okay, now what a trace fossil is, it isn't the fossil remains of or imprint of the organism itself, but of its activity. Okay, for example, footprints, all right? Those are trace fossils, or the tracks left behind by a sea worm crawling across the sediments. Okay, so that's a trace fossil. Or even a burrow of a sea worm, marine worm, that's a trace fossil. And petrification, this can happen in swampy areas, and usually this is with plants, not so much with animals. Well, some of it happens with animals, but with petrification, minerals in the dissolved minerals that happen to be in the water that's moving through the dead organism displace the organic molecules. And in fact, we then have just pure rock. There's no carbon there anymore, no organic materials, but yet the pattern of the minerals follows exactly that of the living organism. And so with some petrified trees, for example, we do cross sections through their trunks and look at them, examine them under the microscope, and you can still see certain features of the cells themselves. That's how good it can be. Another type of fossil is amber. Okay, now what happens with amber, it's mostly a very small animals and plants that will, small organic things that get caught in amber. Amber is originated as resin from a tree. And so even today, when you make a wound on a tree, it'll produce sap. A lot of the sap is very sticky, and it's defensive, it has antibiotics in it. It's a protection against the tree, just like you bleed and clot whenever you get cut. Well, the trees do something similar. And certain types of trees in the past would produce a particular type of sap that would harden, and eventually when it falls to the ground, it stays there for thousands of years as chemical changes, and it becomes amber. Now, any small insect or other organism or seed that happens to fall into the sap sinks into it, gets entombed, and what we have with amber fossils is that the organism is very, very well preserved. As a graduate student at UC Berkeley, I worked with amber fossils that are about 40 million years old from the Dominican Republic, and they were very interesting because I'd take sandpaper and sand it down to about 1 millimeter from the actual organism, the insect that happened to be inside, and polish it and look at it under the microscope, and it looked like very much like an insect that was just out here running around, okay, today. So those are amber fossils. So those are imprint fossils. Here I have a figure from a different textbook that shows various types of fossils. Right here, dinosaur bone, probably a thigh bone, and it's petrified. Okay, it still has some organic material in it, but most of it has been replaced by minerals. And even this human ancestor skull, still most of it is also petrified, it's mineralized. This is a petrified tree trunk. Okay, so there's hardly no organic material there anymore. And this is a scorpion in amber. Okay, I don't know how old this is, but various types of amber can be relatively young to very old. Yes, Alquizim.
So for imprint fossils, does the calcium carbonate cement just fill the imprint that the decomposed organism left?
Yeah. So this is just like if you had mud and you put your hand in the mud, you leave an imprint. You didn't leave your hand.
Okay, so there's no organic material in that?
Well, there is. There can be. There's various carbon compounds that they do find in these, but it's been chemically changed from the type of carbon compounds that you would find in something living. But there's traces of carbon, yes.
Okay.
Is that where like carbon dating comes in order to like?
Not for this old, carbon 14, you can only, and I'll get to that momentarily, you can only use it to date fossils that go back about 50,000 years, 50 million. And that's because carbon 13, the half-life of it is a little over 5,000 years. And so after more than 50,000 years, there's just so little carbon 13 left, it can't be measured. Okay, and I'll get to that here in a moment. But still, radioactive decay of other things, other types of minerals can be used. Emily.
What will we consider dinosaur eggs as then? What kind of fossils?
Those are usually also mineralized. They've been replaced. Yeah, there has been collagen, some types of proteins that have been found in dinosaur bones, but this here would be dinosaur eggs would be like petrification.
Would we consider fossils found in tar pits?
Find what?
Fossils that are found in tar pits, what are those considered? Are those like amber?
Okay, well, it'd be similar to amber, but here they're preserved in tar, and so a lot of times they do have organic, and they're relative. The ones that in LA, those tar pits, these are mammals, and they're recent. Now, when I say recent, I talk about within 50,000 years or so. Actually, 50 million years. Okay, because some of them go back that far, but that tar preserves them, and it is bone that they have there, bone and teeth. And in some cases, if it's a hard part like teeth, like shark's teeth that are found in sedimentary rock, that is enamel. Okay, so it's biological. It hasn't been replaced. But something like here, this petrified tree over here, has very little traces of carbon compounds in it. It's mostly mineral. Okay, and so Alquizim, you have another question in your hand.
Sorry, I'll put it down.
Okay, all right. Now, this is a very interesting, this is also a close-up of an amber insect, and your textbook, 5th edition, and as far back as I know, it still calls this a mosquito. This is not a mosquito. And I don't know how the textbook, no one has pointed out, I'm going to have to do it myself, I guess. This is clearly a winged termite. Now you say termites don't have wings, yet the reproductives do, okay? So the wing venation, I am a wing guy, and sometimes you can bring me an insect wing and without the rest of the body, and I can tell you what it is, all right? This here is not a mosquito, but nonetheless, this amber fossil is about 40 million years old, and it looks a lot of detail there, okay? And you can even have fossils, more recent ones, that are frozen, okay? So here, these tusks, they hook down into the ice into a mammoth, okay? This is not the fossil over here. That's a guy. All right, so fossils. Now, how do we know the age of them? Okay, one that has been built up ever since Hutton's time, mostly during the 19th century by Charles Lyell and others, was what is called the geological column, okay, relative dating. And so using sort of a Linnaean-like classification, they classified various rock strata, okay, layers of sedimentary rock, based upon this principle of superposition. Now, what does it mean? Obviously, if we have rock, sedimentary rock, that has been formed, get the right color here, is that the strata at the bottom, that must be the oldest. And then the next layer was laid on top of it, next layer laid on top of it, next layer laid on top of it. And so as we find these sedimentary strata, these layers of sedimentary rock, the deeper down you go, the older it is. Okay, so they gave names to this, not specific dates. This is relative dating, just saying that certain, for example, Paleozoic strata and the fossils they contain must be older than Mesozoic strata, okay? Simply because Mesozoic rocks are way above and the Paleozoic rocks are further down. And so they would name these into, and they would base, of course, all of the strata of sedimentary rocks are not found in one place on the Earth. Some of it's exposed here and others exposed over there and others exposed over there. Some layers have been missing because the sea in which that rock was being formed over millions of years might have been uplifted, become dry land, and therefore the exposed sedimentary rock was exposed to erosion and destroyed. And then later it was become a sea again, and new layers happened to be up on, lie down on it. And so the way they could put this geological column together, because it doesn't exist in any one place, is to compare and find layers that are in common from site A and site B and site C. And how they matched up those layers is looking for what are called index fossils. Okay, so these index fossils, a combination of certain fossils that indicated that from site A and site B, if they had the same collection of fossils, and that they were the same age, and therefore they could superimpose them together. So let me just jump here, give you an example. Say here, I drew this as site 1 and site 2, and we have index fossils that M and R, that they occur together all the time. And so in stratum B, and at site 2, we also have M and R as index fossils for B. And so we can identify each of these layers based on their fossil fauna, okay, the collection of fossils that happen to be there. Now we notice at site A is that they both have E, but over site 1 we have F, but no F at site 2. And furthermore, but we do have C. So site C, let me get a pin. We have site C here and site C there. Okay, they have the same index fossils. Get to that, okay. And then B, they match. Therefore, if we put these two sites together, that Z and Y must lie obviously between C and B. So why they are missing at C and B is probably after the formation of B, site 2 became dry land, and if there was a Z and Y there, they have been eroded away, okay? Now, if we put these two sites together, we have a geological column such as what I've drawn here, the sequence. And so we're putting these two together. Now, there's a little bit of ambiguity here because we have D at site 2, F at site 1, and we have no information as to which one of these two happens to be the oldest. Okay, so this relative time is simply saying that stratum B is younger than A, that stratum C is older than D, and so forth. And so this is using this principle of deposition to put the fossils together. Now, how do we get absolute age on it? Is if we have some way to radioactively date it. Now, there's in the last 30 years, there's been a lot of different ways to date fossils, and I'm not going to go into too many of them. But if we have right here between C and Z a volcanic eruption, okay, so we have a layer of lava. This here, we can use potassium argon dating to determine its age. Potassium, one radioactive form of potassium will decay into argon. Now, since this was volcanic, heat at very high temperature, when it was laid down, there was no gas in that layer. No argon. Argon is always a gas. And so this would not have had any argon. So once we have that volcanic layer there, and given the half-life of this radioactive form of argon, it would change gradually into, or excuse me, potassium, it gradually will decay into argon. And by using the ratios of the argon gas, and the argon gas is trapped as bubbles in this lava layer, and therefore we can use that ratio, knowing the half-life of it, to determine the rate, determine the absolute age of that radioactive or that volcanic eruption. Then we could estimate, given how thick and how far Z is, we can get an estimate of how old it is. And even Y, we can get an estimate of how old it is. But as we get further and further away from that volcanic layer, our estimates of its age becomes more and more faulty. Okay, there's a greater level of error. Let's go back.
Can you just re-explain why Z and Y were lost again? Was it because C became dry land, or did Z become dry land?
Well, since we still have sedimentary layers here, Y and Z, it's probably at site 2 that it was uplifted, because we don't have Z and Y there, but we have the continuity here of B going to Y and Z. So these have they must have been uplifted if those layers had been there and eroded away.
And how could that happen? Is it, you mentioned going from like a lake basin to like a dry land or something?
Yeah.
Okay.
Yeah, so it's uplifted. If you go out here on Mount Diablo or even Mission Peak, I don't know too much about the geology of Mission Peak, but at Mount Diablo, it's in fact all this mountainous area here in California that once was the bottom of the sea, and there's a lot of sedimentary layers there. And as the Pacific plate has pushed into California or into the North American plate, it's caused all this sea bottom to buckle up, and now it's been uplifted. And so you can go up on Mount Diablo and find fossils of snails, ocean snails. Okay, what are they doing 4,000 feet up in the air? Okay, it's because the land has been shifted up, but it's now open for erosion there, and so those layers are getting eaten away. But of course here, that's eating them sort of sideways, because the rock layers that were laid down flat have been almost uplifted almost 90 degrees. And so as you go over here, you're going back into the past, going the opposite direction, you're going into the future, okay? And so you have these layers across the top. But yeah, they're subject to erosion. Now, so if this uplift at site 2 didn't really tilt the rocks that much, just uplift it high enough out of the sea so that now it's dry land, it gets broken down. That's the fate of all of our land, as it gets eroded eventually.
Okay, I see. And then so why wouldn't a layer like C be eroded after that? What would happen? Wouldn't have to go and do another change?
Yeah, well, it's probable that this, okay, so we're missing Z and Y right here at site 2. So after the formation of Z, this must have been uplifted, and Z and Y have been eroded, and then this was submerged again, became the sea again, so they could accumulate fossils, but it was at a different time span, a much later time period. And so C, we do have them in both places.
Okay, that makes a lot of sense. Thank you.
Yeah, and so this is usually the interpretation of how it goes. So, and I'll show you, in fact, I'll go ahead and do that right now. Let's go here. So this is based on some strata here in the Western United States, the Grand Canyon, Zion National Park, Canyonlands National Park, Mesa Verde, Bryce Canyon. And, you know, not a lot of this, that area throughout much of the Paleozoic and even earlier, okay, because all these, even the Mesozoic was ocean, okay? a shallow sea, and therefore it accumulated these strata. And based on fossils, the fossil collections here, we can put them together. So what's exposed at the top of Grand Canyon is something Calabab formation. We have it over here, and Monocopi formation. We have it here, and the rest of it has been eroded away if it had formed. And so but we can tie together the strata from Grand Canyon and Zion because they have common layers there. And then furthermore, compared canyon lands, there's a lot more exposed here. So we have correspondence in the Monocopi formation, and that formation, Carmel formation, okay, up to there. And then what's in common between canyon lands are these upper formations and the lower formations at Mesa Verde. And therefore, so we can put, if we add all these together, we have what is called the geological column. So the geological column doesn't exist in one place, but in the Western United States, because of the similarity of the fossils, assemblages in these, we have several overlapping layers that correspond from one place to each other, and therefore we can build together a single column. Okay, now the fossils here in Western United States, if we compared them to in Europe, they would not have the same fossil assemblages. And so there's been ways to work around that, is instead of looking at the fossils that show marine layers close to land, it's better to get marine layers deeper in the ocean, because there, within the oceans, most of the organisms, even when they're far, far apart, usually are pretty similar to each other. And so those have been worked out. But just to get the principle here of how it's going, that's what we're talking about. All right, so there's a lot of fossil layers. However, the way in which they've been, so they could talk about them, is to divide them very much like a Linnaean classification into categories, and therefore broadly an eon, and eons, which would be, we're talking about miles of, or almost, well, at least meters of thick rocks that we refer to as eons. And then eons are broken into eras into periods into epochs. Okay, now I'm not going to go down to epochs, but if we go to the oldest eon, this is called the Hadean. Okay, and based on radioactive isotope dating, in fact, Hadean, we don't have any rocks on Earth. Okay, I said that the oldest rocks on Earth are 3.8 billion. But we believe the Earth is 4.6 billion years old. Therefore, we refer to that area, which we have no evidence for, as the Hadean. Okay, so we have no fossil, no information whatsoever, except for from meteorites. And then starting 3.8 billion to 2.5 billion, this is called the rocks, which we do have. We do have some of these exposed. It's called the Archean. And then from 2.5 billion to 0.5 billion, now we're jumping down to 542 million. This is called the Proterozoic. Okay, now in older textbooks, these older areas from 4.6 to 542 million years ago are often called Precambrian. Now, why are they called Precambrian? Because the first era, or not the first era, the first period in the Phanerozoic, which is our recent time from 5.4 billion to today, that is referred to as the Eon Phanerozoic. The first period in the Phanerozoic is called the Cambrian. And you don't need to know that, but they called all this area back in the 1800s, called all this area before 0.5 billion years ago, the Precambrian. I don't want you to use this. I really want you to refer to Hadrian, Archean, Proterozoic, Phanerozoic. These are eons. And I'm not going to worry about the breaking them down into eras or periods, except for the Phanerozoic. The Phanerozoic is famously broken into 3 eras. the Paleozoic. And these are based primarily, now what are the boundaries between these? There's geological events that mark the boundaries between these various eras, and we'll talk about it. So the oldest is Paleozoic, and Zoic refers to zoo, okay, animals, right? Because these have mostly been put together in the 1800s on the basis of animal fossils, and Paleo meaning old animal life. And Mesozoic, middle animal life, and Cenozoic is where we are now, is modern animal life. And as we go look at the animal fossils throughout the Phanerozoic, the Paleozoic fossils look very, very different from anything that we have here today. And as we move down through Mesozoic into Cenozoic, we become more and more similar to what we have, that we see around us today. So Paleozoic, from about 542 million to 251 million years ago. And there was an event that marks the end of the Paleozoic and the beginning of the Mesozoic. The Mesozoic has 3 periods that I do want you to know because they're pretty common. This was the age when the dinosaurs were around, was in the Mesozoic. And there's 3 different periods there. And this is the only one I want you to know periods for, and you don't need to know when they began or when they ended, but just only the sequence. The oldest is Triassic. And most of these things you've heard before. If anyone's paid attention to dinosaur or ancient reptile fossils, there's the Triassic, and then the Jurassic, okay, Jurassic Park, you know, that's really the high time for the dinosaurs. And then the Cretaceous, all right? And then after that, and there was an event that we'll talk about that marks the ending of the Mesozoic and the beginning of the Cenozoic. The Cenozoic is from 65 million to today. And although it's also divided up into into periods and epochs, we're not going to worry about those, all right? But I do want you to know these are approximate dates, but definitely the sequence in time. Now to give you some idea here about, okay, we've already gone over this. Okay, this is in your textbook, a table that's mentioning these eons, Hadean, Archean, Proterozoic, those are eons. And then Phanerozoic is an eon. And then within the eons, there's eras, Paleozoic, Mesozoic, and Cenozoic. And then those are divided into periods. And although I will be using in my presentations I go on here, I will be mentioning these periods, you don't really have to know them. I just want you to know the eras and eons, except for the Mesozoic. All right, now this sort of gives you an impression, wow, there's a lot of stuff that's happening in the Phanerozoic, and this gives you an impression that the Phanerozoic is a long period of time. That's actually wrong. Most of Earth's history lies here, okay, in these places. What is so important about the Phanerozoic is this is an era in which fossils start to become abundant. In Darwin's day, they didn't have any fossils back here. All of a sudden, about 540 million years ago, we start to have an abundance of animal fossils, and they appeared to show up rather suddenly. Now, since then, we have found fossils in these other rocks, but they're hard to come by. These other rocks here are very, very, very old. To give you a more realistic time frame, let's take all of Earth's history and put it into 1 year, okay? So that the formation of the Earth was on January midnight, okay? So this January 1st, it's formation of the Earth. We don't have any fossils. Our first, well, actually, these are hypothetical. We believe that the first cells, all based on DNA sequences, probably showed up about, almost 2 months later after the formation of the Earth. And our oldest rocks that are presently on Earth is not until March 4th. And then the first fossils of bacteria, 20th of March. And eukaryotic cells, not until July, okay? And the beginning of the Paleozoic with Cambrian explosion, that's way into December, okay? So this is when animal fossils start to become really abundant, not until November. And then the first insects, first amphibians, and so forth, first flowering plants, 22nd of December. This begins the Cenozoic, the area we're in now, 26th of December. And humans, anatomically modern humans, Homo sapiens, doesn't show up until December 31st at 11:48 p.m. Just and the Revolutionary War just only happened 1 second ago. Okay, this gives you a better idea as to the dimensions of time with these eras. Okay. All right, so the absolute dating of fossils. There's many different dating techniques available today, but one of the best ones is the use of radioactive isotopes. These are unstable so that they decay. Carbon-14, it's already been mentioned. It's a good for dating organic materials such as bone and hair and teeth and wood from trees. However, it has a very short half-life. Now, this carbon-14 is formed high in the atmosphere. Actually, I think it's from a form of nitrogen-14 and UV light, but it creates carbon-14 that has a half-life of 5,750 years. Now, what does a half-life mean? It means that when you have a certain amount of it present at birth, because living organisms take in carbon compounds, all right? You eat, and that carbon, a lot of it is carbon-12, but still there is carbon-14 that's in there. And so your bones and teeth will accumulate this during your lifetime. Now, once you die, that accumulation stops. And so once that accumulation stops, then our carbon decays, all right? So a half-life is the amount of time it takes for 1/2 of that material to disappear. So if we started out with a sample and we had 10 milligrams of carbon-14, it's more sensible to make that micrograms. Then after 1 half-life, 5,730 years, there will only be 5 milligrams of, actually micrograms, of the carbon-14 left. And then after another 5,730 years, 2 half-lives, then that's how long it takes half of the 5 milligrams to disappear. So that's now 2.5 micrograms and so forth, okay? And so that is the half-life. In chemistry and physics, they would probably give you a particular exponential formula so you can count how old something is based on the data. But here in this class, I'm just going to stick to half-lives. And so if 2/3, or excuse me, 3/4 of the material had disappeared, 2 half-lives have passed. Okay. Now, one of the problems with carbon-14 is that it's in small abundance. So a bone wouldn't, at death, wouldn't have a whole lot of it compared to carbon-12. It's probably only less than, it's actually less than 1%. And so after several half-lives, you know, there's not going to be too much of that carbon-14 there, and it gets to the point where it can't be measured anymore. And so carbon-14 as a tool for absolute dating is only good for about 50,000 years. Beyond that, we can't date anything. Okay. Now, remember that dinosaurs, they disappeared, went extinct 65 million years ago. Okay. So 50,000 years is not a very long time span. And furthermore, we know that the levels of carbon-14 that form in the atmosphere is not constant over time. And so at certain times in the past, there was higher amounts of carbon-14 being formed than at other times. And therefore, we have to calibrate the carbon-14 clock. The way they do it is by something called dendrochronology. Some of our trees are very, very old, okay? And so if we have a tree, and of course, when the trees lay down rings in the wood every year. We have some redwood trees that live for about 2,000 years. And furthermore, there's carbon for this year, carbon 14 for that year, and so forth. And so scientists can take a core out of this wood and measure the carbon 14 content by each year. And knowing the decay rate, half-life of carbon 14, they can calibrate and figure out how much carbon 14 had to be in the atmosphere during that year. Okay, so this tree might have 2,000 to 2,500 rings. Okay, so it spans over that time. Now, how can we go back further in time? We have fossil trees. We have a fossil tree that died some time ago. But if it has tree rings that overlap with some of the younger, okay, some of the old, this is newer tree. This goes from 0, this is alive today, and its innermost ring, let's say it's 2,500 years ago. We have to count from the outside toward the middle. And here we have another tree. Let's say that some of the first 500 years of our present tree corresponds with the outer rings of a fossil tree. So these are the same, and this can push the chronology back another, this goes from 2,500, no, 2,000 to 4,000 years ago. Now, how can they match these rings? The rings are not the same width from one year to the other. It depends on rainfall. Okay, so if there's a drought, then you'll have a ring that is very, very thin. And if that drought lasted for 5 years, you would have 5 very, very thin rings. And then if the drought gets over and we have a lot of rain, then we have thicker bands. And therefore, due to the width of these rings, we can, if they overlap, we can join them together and we can extend time back. Furthermore, since we can date these wood, each layer of the wood, we can determine the carbon 14 content that had to be in the atmosphere based on the carbon 14 content that we have left. Yes, Alquazim.
How would this fossil tree have to be like preserved to maintain like organic material?
It would have to be wood, you know, and some of this wood that doesn't decay. And so the dendrochronology, we actually have wood that is still organic. Okay, it's still cellulose and lignin even after 50,000 years. Okay. So we have been able to calibrate it back that far, but not any further than that.
So it's still stuck to that 50K limit.
Yeah.
Okay.
Yeah. Okay, so this isn't good. So this is good. Carbon 14 is good for something recent, but there are other radioactive compounds I already mentioned from volcanic eruptions as potassium 40. It decays to argon 40. And so if we have a volcanic eruption, we know even if there was argon gas there to begin with, it escapes because that rock is so hot, gases aren't there. So we can pretty well know that lava flow, that volcano flow, it started out with just only potassium 40, but no argon. And therefore, this has a half-life of 1.3 billion years. And so if we come and we measure that there's 50% potassium 40 and 50%, there's a 1-to-1 ratio between the potassium and the argon, then that's 1 half-life. Okay, so it's 1.3 billion years old, and so forth. Okay, and so this is very useful for older things. And there's also uranium isotopes that can be used as well, and I'm not going to go into them. But nonetheless, this absolute dating is based on finding something that is datable within the geological column. So there's 2 ways of looking at this: absolute time and relative time. Okay, this is where I'm going to stop. Didn't get very far. What I'm going to do is start with the Archean. Well, actually, we don't need to say anything about it because that's just an era, eon, that we put there to account for the oldest rocks. And so it ends, or actually the Hadrian. I'm not going to talk about the Hadrian. So I'm just going to talk about what fossils do we have down through life and what can we say about how things have changed. And that's where I'm going, and I'll go further into that on Tuesday. So any questions?
Can we go to slide 15?
Slide 15.
Thanks.
Okay, anything else? Okay, if not, then that's where I'll quit.